about two-photon optogenetics. Advance of optogenetic methods provide an unprecedented toolkit for modulating brain circuit with light. For the light-based regulation of gene expression, synaptic release, and biochemical signaling pathway optogenetics technique have been created. Proteins known as optogenetic actuators alter the activity of cells when cells are exposed to light, which can be used to create single or many action potentials, inhibit neural activity, or alter biochemical signaling pathway. Opsins are the most potent and often utilized actuator. Short blue light pulse that activate CHR2 excitation can be used to trigger individual spike, and consecutive yellow light pulse can be used to block those spike, activating NPHR inhibition. The response to visual stimuli is significantly influenced by the neural excitability, which fluctuates over time and space, and which either excites or inhibits large population of neurons. Therefore, to provide insight of neuroscience concerns, such as the relationship between the neural code and behavior, advancements in Neurophotonics techniques with higher precision and built-in optical readout capabilities are needed. Holographic illumination has a number of advantages over other light patterning techniques, including a relatively large excitation field of view, the ability to generate a variety of illumination patterns, and to dynamically correct the projected pattern. It also offers high efficiency modulation. SLMs are optical components that modulate the path of light as it travels through space and time by just changing the phase of the inside light, making them more power efficiency. Another significant benefit is the capacity to create three-dimensional light pattern. As a result, optogenetic two-photon excitation has emerged as the primary technique for accurately activating neurons in brain tissue. Holographic photon excitation must overcome significant difficulties to provide more effective stimulation. First, while avoiding tissue heating, high laser power and good power efficiency are necessary in order to improve the likelihood of excitation. Second, because the signal is now proportionally to I squared rather than I, two-photon excitation enhances the speckle contrast, that is, the speckle fluctuation. So fluctuations should be reduced. The tiny excitation region of two-photon excitation is likely the third and biggest hurdle.
Since only small patches of the target neuron cell membrane are affected, tightly focused diffraction-limited focal volume are not very effective at changing the behavior of the target neurons. The two general strategies of defocusing the focal spot to cover the entire cell body, or fast scanning a diffraction-limited spot to cover a significant portion of the cell's membrane, are typically used in the practical implementation of two-photon optogenetics. The first method sacrifices temporal resolution needed to create precise spike timing and reproduce spatio-temporal pattern activity. The second strategy calls for more complex hardware. By modifying the spatio-temporal profile of the light pulse, temporally focused excitation is employed to distinguish the lateral and axial dimensions of the excitation area. Optical harness technology is a very active area of neurophotonics, and numerous recent studies have improved both the spatio-temporal properties of the produced light patterns and essential performance parameters of the optogenetic probes. For this use, several new families of redshifted optogenetic probes have been created. Soma targeting techniques have also been developed for enhanced efficacy and precision. and transgenic animals that express these probes under genetic control have been developed. As a result of their longer excitation shoulder at lower wavelengths, these opsins suffer from undesirable crosstalk excitation from the two-photon imaging laser, despite having the advantageous attribute of being efficiently stimulated at the 1040 nanometer wavelengths. Combining a red-shifted indication with a blue-shifted opsins is one potential alternate approach being researched. Early attempts to use precision optogenetics in awake behaving animals have not been able to produce any detectable behavioral consequences. Several approaches have so far been developed to deal with this problem. The first is to make the manipulation more intense, for instance by increasing duration. Second, the animal is trained to become more sensitive to minor disturbance by lowering its perceptual threshold by several orders of magnitude. Recent research on the coding logic of olfactory and visual perception is supported by these methodologies. Here's our references. Thank you for listening.